Good morning, everyone. Um, uh, it seems that some people are finding reef problems. Nobody has. Let me just see if I can run a quick one. Can anybody vote? Does anybody see that that reef is open? Not connecting. Does that mean you can't enter the class? All right. Let me see if I can reload it. Let's see if that makes a difference. Hold on a second. Oh, I got a couple of votes. I have 2 people have joined. You want to try again? Can anybody else vote? I've got 2 people who have voted. Unable. That's weird. All right, give me a second. Let's see. Hold on. I'm going to close it and then open it again. Okay, I have started a new session. Okay, can everybody make sure that you're muted? I see someone has their mic open. Okay, can everybody make sure the mic open? Thank you. All right, you guys can see me? People been able to join? Okay, good. All right, we're in action. Okay, so we are looking at our cytoskeleton. We are going to continue looking at microtubules, some of the functions of microtubules. I've got a couple of other nice animations from SmartWork that I think are valuable. I think I'll be able to show one of them today, and we'll start talking about muscle contraction, but probably won't finish that until Wednesday. So make sure that you have joined your reef. Looks like everybody can now. Sign into your reef account and join our course. And we will pick up where we left off last week, where we had been describing microtubules as one of the types, our little green filaments here that we had described last week are dynamically unstable, meaning that individual microtubules can grow at their plus end, but they can also shrink at their plus end. Now, some of those microtubules may be stabilized if they encounter a protein at some part in the cell. That protein may prevent that GTP cap from essentially capture the GTP cap and keep that microtubule stabilized such that it can't shrink. And when that occurs, a number of microtubules may be stabilized and may maintain their particular length, won't be able to shrink anymore. And this will contribute to what we call polarization of the cell, where one side of the cell has different structures than the other side of the cell. And that also means that one side of the cell will have different functions from other side of the cell. Sometimes if we're looking at cells that are secreting, they may have the Golgi sort of oriented on one side of the cell so that they secrete, they'll release their vesicles to one end. Sometimes a cell that's migrating will be polarized. One side of the cell will be able to move, extend regions of the plasma membrane move in one direction versus another. But one highly polarized cell is a cell that we have already been spending some time looking at. A neuron is an example of a highly polarized cell, where one end of the cell is receiving information and the other end of the cell, the axon terminal, that can be very far away if that axon is very long, will be communicating with a downstream, with another cell. And if we sort of look at that axon, we see that the axon is packed with microtubules. The axon we already described is also packed with neurofilaments. Those would be intermediate filaments that are giving some structure, helping to give some support to this very thin but long axon. But what we're focusing on here is our microtubules. Note that there would be a number of parallel microtubules that are stabilized. They will, once they've grown, as the axon has grown towards its target cell, with the plus end at the axon terminal end, these microtubules won't be shrinking anymore. There will be some other proteins that maintain that elongated state. And what is the role of microtubules in an axon? This is where microtubules serve as a track that vesicles will travel on. Vesicles, for example, secretory vesicles that were shown here in red, that are carrying that neurotransmitter to the axon terminal. Remember, this is a form of regulated secretion. As soon as, because this is in the cell body, there's the nucleus, this is where the ER would be, this is where the Golgi would reside in the cell body of a neuron, and all of those neurotransmitters will be carried in these secretory vesicles to the axon terminal, where they will wait until they receive a signal, the signal for the secretory vesicles to fuse with the plasma membrane and then release their contents into that synaptic cleft. Right, we'll look at that a little bit later. What is that signal? But in order for them to get there, they have to travel along these tracks. And these vesicles are traveling along the tracks because they are bound to some proteins that we call motor proteins. The motor proteins are actually moving. They are transporting vesicles. And there's a number of different types of motor proteins. And we're going to spend some time looking at them. Note that the motor proteins actually move these vesicles fairly slowly. It could take maybe a day, maybe even a week, for a secretory vesicle to travel down a track to get to the end of a very long axon. But that still would be faster than just simple diffusion of these vesicles throughout the cytoplasm. So we want to spend a little bit of time looking at what is a motor protein, and then see how particular motor proteins interact with microtubule tracks. So a motor protein is a protein that can move. If we look at the structure of a motor protein, and that we'll see on the next slide, there is a head domain, which is the domain that's actually doing the moving, walking along the microtubule. And there is a tail domain that will attach to transmembrane proteins on a vesicle, in the membrane of a vesicle that is carrying material throughout the cell. And so the movement of these vesicles or organelles, pieces of Golgi, pieces of endoplasmic reticulum, parts of a cell that have to move around, let's say as a cell is undergoing cell division, move it to distribute it throughout the cell. This movement, again, this is not just Brownian motion, it's not just diffusion, it's an energy dependent process, requires energy. So we've got a couple of different families of motor proteins. Dyneins are motor proteins that move towards the minus end of a microtubule, so they're directional. And kinesins are motor proteins that move towards the plus end of microtubule. So if we look at examples of these two different types of motor proteins, we will see that each of them they're slightly different shapes, different types. They bind to different types of cargo, and they move in particular directions in the way that they interact with that microtubule. Because of course our microtubule has a polarity, right? A plus end and a minus end. Kinesins will only walk towards the plus end. The head portion is the ATPase domain. This is the portion of, and again, both of these are dimers, right? So you have two individual kinesin or polypeptides that make up a kinesin. The head domain will be able to bind ATP, and it will also hydrolyze ATP. The same thing with a dynein. A dimer, the head domains are the parts that interact with the microtubule. They are ATP binding domains. The other end that we call the tail end is the part that will interact with the cargo, and that whatever is being carried around, whatever is being moved in the cell, that cargo, again, could be a vesicle full of neurotransmitter or other types of products that will be secreted. The cargo could be an organelle or a part of an organelle. And these head domains will walk, will move in a characteristic manner. One domain change its shape, interact with our microtubule. The other domain change its shape and move in one direction, either towards the plus end or towards the minus end, depending on what the motor protein is. So let's look a little bit more carefully at where is the how does this walking occur, and where does ATP come into play? And this slide here shows it a little bit better. If we're looking at our green little blob here is our motor protein, interacting with, in blue is our microtubule track. What we have is a series of shape changes to our motor protein. When ATP binds to that motor protein, it causes one type of shape change such that a region of that motor protein interacts with the microtubule. And then in the next step, the ATP is hydrolyzed because the motor protein itself is an ATPase. It can bind ATP, it can hydrolyze ATP. That hydrolysis causes a different change in shape of the motor protein. And then the ADP and inorganic phosphate products of that hydrolysis will be released from the motor protein, and that causes a return to the original shape. Right? This sequence of events of binding, hydrolysis, and release over and over again causes these repeated shape changes that results in directional movement. Right? So yes, and every time it takes a step, so that this is going to require a huge amount of ATP. The sequence of events, one individual molecule, one individual motor protein, which is a dimer, will carry out these shape changes. All right, so in this little cartoon here, what this is showing is that not only do we have these two different families of motor proteins, and we describe members of these motor proteins. They have similar structures and similar functions in that all kinesins, again, move only in the direction towards the plus end. All dyneins always move towards the minus end. What this figure is trying to show is that different types of kinesins have different domains on their tail end that will allow them to bind to different types of cargo. So some cargo moving to the plus end, that type of cargo would often be involved in exocytosis because our plus ends of microtubules always extend towards the periphery of a cell. So kinesins will be carrying cargo, would be involved in exocytosis, where dyneins would be involved in endocytosis, carrying cargo, let's say vesicles and an endocytic vesicle picking up food from the plasma membrane and carrying it towards the interior of the cell. All right, so what are the kinds of things that we have described so far about our microtubules and our microtubules interaction with motor proteins? Here's our first question. Oh, I'll go to this in a second. I'll come back. Let's answer our question first, and then I'll come back to our little video. Someone's asking what happens to motor proteins getting to the end. The motor proteins might then become, I don't know, they probably would become degraded, may depolymerize. They may be part of a pool that has to then reform because they can't go backwards. That's a good question. Do they walk off the end? That I am not sure about. Seems like they would have to. How's everybody doing? So you're reading our four statements, and only one of them is correct. So think about what's wrong with the other statements. All right, how about 5 more seconds? All right, let me close it up. All right, looks like the popular answer would be D, and D is the correct answer. These motor proteins that we've just described move directionally, right? Because they interact with that microtubule, all those individual protofilaments that make up that cylinder, that has a polarity as well. So they interact with the tubulin heterodimers in specific ways directionally. So that's correct. A is not correct. Microtubules of a spindle, or really any microtubules, originate at the centrosome, right? The microtubule organizing center is a centrosome, not a centromere. Microtubules, some are unstable, but yes, but some are stable. And for example, these tracks that we've described, those would be stable forms. And we'll look at another example of microtubule functions in cell, stable filaments that don't shrink. Once they've grown to their final length, they don't shrink again. And as we know, in tubulin, microtubule formation, the tubulin heterodimers bind to GTP, not ATP. So microtubules are also present and function in particular cell types, cells that have cilia. And this would be an example of an epithelial cell that one side has extensions of the plasma membrane. And those extensions are not only stabilized by the presence of microtubules that have grown, and again, a basal body is another example of a microtubule organizing center that microtubules grow out from. So the plus end would be at the sort of the tips of each of these cilia. And these microtubules can move. The movement of the microtubules allows the cilia to move, the cilia to have a sort of either wave-like, or in the case of cilia, sort of a rowing motion. And cilia also, in some cells, have a similar structure, which is a flagella, a flagellum, a single flagellum, which again is a long extension from the cell. So we're going to look at how it is that microtubules can cause or drive this type of movement of cilia or flagella. As an example, the roles of cilia in some cells for unicellular eukaryotes, cilia allow that, let's say a paramecium, to be able to move. All that beating motion of all of those individual cilia on its surface allow it to be propelled through its aqueous environment. Some multicellular eukaryotes, like ourselves, we have cilia on some cell types, such as the cells of our respiratory tract, the cells in our trachea and our bronchial tubes that are constantly moving. That beating motion will cause movement of the fluid containing mucus as well as debris in our respiratory tract and move it up towards our throat so that we can swallow it to keep our airway clear. And then sperm cells have a similar structure, a single flagellum that has an undulating motion as opposed to a rowing motion of cilia, an undulating motion, a wave-like motion that allows that sperm cell to be propelled. So how do those structures allow for movement of these cells or movement of fluids across the cells, like in the case of the ciliated tracheal cells? If we look at a cross section of a cilium or a flagellum, we would see that they have this characteristic structure, this characteristic arrangement of microtubules that we call the axoneme. Microtubules that are arranged in doublets, 9 of them around the periphery, and again, within, covered by our plasma membrane, and 2 single microtubules in the center. So we call this the 9+2 array of microtubules that make up an axoneme, which also is associated with some other proteins. A motor protein, specific motor protein, dynein. So what we'll see is these red structures are indicating the head domains of dynein motor protein that's attached to one of the microtubules of the doublet. And what we're going to see is that dynein, they're calling it here the arm. We could also think of it as the head because it's the ATP binding domain. Will also interact with the one of the microtubules of the neighboring doublet. We'll see this a little bit better if we look at a longitudinal arrangement. And so this doublet, one doublet is going to be interacting with the other doublet, its neighboring doublet, via motor proteins. But equally important is this blue protein that is a linking protein that we call nexin. And nexin is going to limit the type of movement of doublets relative to each other. All right, so we're just describing the structure here, which we have elaborated here. Dynein is the motor protein. The tail's attached to one tubule, and the head domain contacts the other microtubule, the adjacent microtubule. And the nexin is the linking protein. The dynein is going to be walking along one doublet and really carrying one microtubule relative to its neighboring microtubules. So let's take a look on our next slide. Here we have isolated a couple of, or a collection of microtubules. So we have one doublet here on the left-hand side and another doublet on the right-hand side. Notice the orientation of our microtubules. Our plus end is up, our minus end is down. And the red is showing us our motor protein, our dynein. So here is the tail that is attached to one microtubule of the left doublet, And the head domains, these are the ATP binding domains, right? These are the parts that actually move, are attached to one of the doublets of our one microtubule of our right-hand doublet. If we add ATP to this solution, what we'll see is all of those head domains will interact with ATP in that directional manner, and they will start moving towards the minus end. That results in this left doublet moving, right, towards the bottom in this arrangement. The left doublet is moving towards the minus direction relative to the right doublet. And so they're sliding relative to each other. But in an actual cell, those two doublets are attached to each other by our Nexin linking protein. And so even though a force will be applied in the presence of ATP, where these ATP binding heads will be moving towards the minus end of this right-hand doublet, that force cannot result in sliding because the doublets can't slide relative to each other, and the force is then converted into a bending. So the presence of the linking proteins causes the doublets to bend relative to each other instead of sliding relative to each other. And that's just showing this little bit of this bend in this undulating motion. And remember, we actually have our axoneme, right? We have these doublets all in a circular arrangement. So depending on which dyneins are walking relative to which doublets will determine which kind of bending is occurring, right? Bending to the left, bending to the right, in this undulating motion of this flagella of this sperm cell. So I have a question for you. Here we're looking at an example of what we just described. We have our two doublets of microtubules. We have our linking proteins. We have our dynein motor proteins. And what we're asking is, what if the polarity of those microtubules were reversed from what we had just described? What would happen in that situation? We add ATP. What kind of movement would occur? Oh, yes, I forgot the video. Yes, after we answer this one, I'll put it up. Thank you for reminding me. How's everybody doing? How about 5 more seconds? All right, let me close it up. Everybody's in? All right, closing it up. And the correct answer would be C. If we have, if we're really just looking at our doublets where we have the minus end up, then in the presence of ATP, all the dynein motor protein, the ATP binding heads would be walking in this direction, right? Walking towards the minus end of the right hand doublet, which would result in bending towards the left, right? They can't slide relative to each other, but we would have our bending towards the left instead of to the right. Right? We're trying to emphasize it's that directional movement that causes this directional movement of the motor protein that causes this directional change in the shape of our microtubule doublets. Okay, All right, so let me get up our little video. Hold on a second, I gotta change, let's see. Yeah, there's audio on this one too. So hold on a second. Close this down. Fix the audio. Okay.
In this experiment, a cell homogenate containing many different organelles is added to microtubules. Motor proteins are normally attached to the organelles. When ATP is added as a fuel for the motor proteins, some organelles bind microtubules and are moved along the tracks by their motors. Most kinesin motors move towards the plus end of microtubules. Dynein motors always move in the opposite direction. Both motors are used to transport organelles, and occasionally a single organelle, which must have both types of motor attached, can be seen to switch directions. The bidirectional traffic observed here is reminiscent of that in an intact cell.
Okay, so that's kind of a nice demonstration, a little bit nicer than all of those just looking at cartoons. All right, let me fix my audio again. All right, so I hope you thought that was worth taking a peek at. Again, those individual sort of microscopic either images or videos, I think, are really valuable to get a better flavor. For getting a better flavor of what's really, especially when we're looking at movements or changes. So now we're going to move on to our third class of cytoskeletal filaments, the smallest group of microfilaments that we call actin filaments. Actin filaments are also called microfilaments. They're a type of cytoskeletal filament, the smallest ones, the thinnest, the smallest in diameter, about 7 nanometers across. And we find actin filaments or microfilaments often in the periphery of a cell. Their primary role is helping to maintain a shape of a cell or to cause the shape of a cell to change. So over here on the left-hand side, we're looking at an example of a role of microfilaments, is that microfilaments help to stabilize the shape of microvilli. These are tiny little extensions of the plasma membrane. This is not a cilium. A microvillus is an extension held out from one end of our cell by bundles of actin filaments. And really, this is just expanding the the surface area of the plasma membrane. So a cell that's going to be absorbing, let's say, cells lining our gut, absorbing nutrients, need more surface area. And so microvilli have a role in just expanding the surface area of plasma membrane. And this shape of the cell is maintained by actin filaments. Other types of cells, their shape, again, is maintained by the arrangement of microfilaments. And if this cell's shape is going to change, that change could be manifested by changes in the lengths and the arrangement of microfilaments. In C, we're looking at a cell that's moving. This broad sort of leading edge of the cell, this cell is migrating sort of to the position of about 5 o'clock. Little finger-like projections called philopodia have collections of actin filaments that are all growing in the same direction as that cell is migrating across the surface. And microfilaments are also involved in cell division. Microfilaments will interact with a motor protein, a microfilament specific motor protein, during cytokinesis to sort of split that dividing cell into those two cells, right? Sort of pinch off at the midline. So let's take a look at these actin filaments, these microfilaments, and see what they're made of, what are the subunits, and then how do they work. The individual subunits are globular proteins, so we call them G-actin. G-actin monomers can bind ATP. They're also polarized. They have a plus end and a minus end. And they assemble into these long arrangements of these G-actin subunits into a filament. And so this polymer of actin monomers we call F-actin or filamentous actin to distinguish it from the subunits. So the individual polypeptides are called G-actin. When they're assembled into filaments, we call them F-actin. And so here's the individual subunits here. They will associate in a characteristic manner. The plus end, which we haven't indicated plus and minus here, but plus end of one monomer then interacts with minus end of another monomer, and they are rotated 160 degrees relative to each other, resulting in an overall helical arrangement of these polymers. And the actual F-actin is essentially a double helix of chains of G-actins with a plus end and a minus end, right? So the polymer, that's double helical polymer, we call F-actin. Now, F-actin can grow, and actually it's very similar to microtubules. It's possible for F-actin to grow at either end, but it primarily grows at its plus end by the addition of more G-actin subunits. Primarily, G-actin that is bound to an ATP will associate with the plus end. This will be growth or extension of this filamentous actin. The ATP is then hydrolyzed to ADP, so that G-actin is an ATPase. This is sounding very familiar, right, to how the subunits of microtubules add and can hydrolyze their bound nucleotide. And a G-actin that is bound to an ADP does not have the same affinity for other monomers, and so tends to dissociate. And so if you actually put together, either in a region of the cell, or you could do this experimentally, if you had a constant amount of G-actin monomers, and you had constant association rate at the plus end, so this actin bound to ATP adding one at a time to the plus end, which will relatively quickly hydrolyze that ATP so that now we have it pink. The bound nucleotide is now ADP, which has less affinity. So if we had a constant addition of ATP bound actin at one end and a constant dissociation rate of falling off of actin bound to ADP, which can then exchange its ADP for ATP in the pool in the cytoplasm, you would overall have what we call this treadmilling, right? Constant addition at one end, loss at the opposite end. The actin filament length would stay the same overall, but it would be moving to the right side of this slide, right? So if there's a number of microfilaments doing the same thing, adding at one end, losing their subunits at the other end, you would have a collection of actin molecules moving to one end which could result in the cell moving to that side, right? That end of the cell could be extending. And that's one way that, one part of the migration process of cells that do migrate across surfaces. But not all actin filaments grow at one end and lose. Some actin filaments can be stabilized. There are a number of different types of accessory proteins that interact with these different types of filaments, whether we're talking about microtubules or if we're talking about microfilaments that will stabilize the length of this cytoskeletal filament. And we're going to look at an example next of a actin microfilament that has a stable length and that also interacts with a specific type of motor protein that will result in movement of a cell. And so what we're looking at then is the cytoskeletal components of muscle. Here on the left-hand side, we're looking at a muscle cell, which we call a muscle fiber, because a differentiated functional muscle cell is actually formed from the fusion of a number of myoblasts, young undifferentiated cells fused together to form this giant cell, which contains a number of nuclei. And this muscle fiber is packed with these bundles of protein that we call myofibrils. Now, if we look at a myofibril, what we see is that myofibril, again, is packed with all of these parallel arrangements of cytoskeletal filaments. The cytoskeletal filaments, here they are all arranged parallel to each other. We see within the filaments are composed of these functional units, contractile units that we call sarcomeres. The sarcomere is the unit that will actually change its shape. It can contract, and all of these sarcomeres among all of the myofibrils contracting together, changing the shape or changing the arrangement of all of these parallel cytoskeletal components relative to each other results in contraction of this muscle cell. So the next slide, we're going to blow up an individual sarcomere, the contractile unit. What are sarcomere, and why this is why skeletal muscle cells look striated, they look like they have stripes due to the arrangement of all of these parallel filaments within each of these individual sarcomeres. The filaments of the sarcomere are thin filament is our actin filament, here shown in pink. Sort of interdigitated with thick filaments that we show here in green, which we call a myosin filament. So myosin filaments and actin filaments, we can actually see in the sarcomere, giving sort of different degrees of density. The thick filaments are giving this sort of darker density, thicker density in the center of a sarcomere. The individual actin filaments are attached to some proteins that together we call the Z disc. So these are stable microfilaments. Once they have grown, they grow such that their plus end is attached to the Z disc. So the plus end here, plus end there, and their minus end faces the midline of the sarcomere. Okay, the minus end is pointing towards the center. So how does this arrangement of these parallel filaments result in contraction? Well, again, the myosin thick filament is made up of motor proteins. Myosin is a motor protein. Myosin moving, walking along actin filaments results in overlapping of these filaments relative to each other. The myosin is the motor protein, an actin-dependent motor protein that interacts with ATP, hydrolyzes ATP, releases ADP, right, in a sequential manner that allows the myosin heads to walk along the actin. And so what we have is, if we sort of blow up again our sarcomere, and I'm going to add the orientation of our filaments. Here's the plus end is attached to the Z disc. All of these thin filaments, all of these actin filaments have their minus end facing towards the center of the sarcomere. When that sarcomere is induced to contract, what that means is all of those individual myosin heads, and this myosin filament is sort of like a double-headed arrow. All of the heads of the individual myosin motor proteins are going to move directionally towards the plus end. All of these heads, and there's about 300 myosin molecules per myosin filament. All of these heads on the right are going to walk towards this end, the plus end, and all of these heads are going to walk to the plus end of that actin filament, such that they will force, by them walking, the myosin heads walking in opposite directions, they end up forcing the actin filaments towards the center, increasing the degree of overlap, shortening the sarcomere overall, which results in contraction. The relaxation is when the myosin heads let go of the actin filaments, and then the sarcomere returns to its original position. So what are all of these motor proteins made of? What do they look like? How do they interact with the actin filament? That's what we're going to look at next. If we looked at just one myosin motor protein, this is actually myosin 2. There's different types of myosin motor proteins that function in different cell types. Many cells that aren't muscle cells have some cell movement or some degree of contraction. So different types of myosins are expressed in different types of cells, the myosins interacting with microfilaments in different ways. In the case of muscle cells, we're looking at myosin 2, and myosin 2 consists of a globular head region. This is the region of this polypeptide that interacts with ATP, and the polypeptide then extends into this long rod-like domain. Two myosin 2 molecules, so this is a dimer, will interact with each other, forming this very long tail domain. And then the tail domain of one myosin dimer will interact with the tail domain of another myosin dimer, right, in the other direction. So you end up with this double-headed filament, right? Couple hundred myosin heads at one end, a couple hundred myosin heads at the opposite end. There's also other regulatory protein called a myosin light chain. We don't need to worry about that. That's going to regulate how these head domains move. But we're just going to really focus on thinking about the overall shape and the arrangement of these myosin molecules in that myosin filament. And then we're going to look at how ATP interacting with this head domain affects how the myosin motor protein interacts with the actin filament to cause this movement, this sliding of filaments. And that's what we're going to look at the next couple slides. Described here, shown in our next couple of slides. We start with our we're just following one myosin head of the several hundred that's extending out of our myosin filament. It starts by this is a cycle. And at the beginning of the cycle, the myosin head is attached to one of the subunits of our actin filament. And here's our plus end to the right. In the presence of ATP, ATP binding to that myosin head causes the myosin head to release, to detach from the actin subunit. The ATP will then be hydrolyzed because this head is an ATPase, has ATP's activity. The hydrolysis of ATP to ADP plus inorganic phosphate causes a change in the shape of that myosin head so that it moves, excuse me, it moves relative to the actin filament a little bit closer to the plus end. It moves from a position close to this subunit to close to the next subunit. We say that's in its cocked position. Very quickly, the inorganic phosphate is released from the myosin head, which allows the head to bind to the actin subunit, which causes the release of ADP, and the release of ADP causes what we call the power stroke, the change in shape back to the original shape. That power stroke has now caused the myosin filament to move to the right towards the plus end, which is in effect moving the actin filament towards the left to the minus end, right? So repeated. Then the next time, right, that myosin head is attached to actin. Same thing's going to happen in the presence of ATP. It lets go, it hydrolyzes, it moves to the cocked position. It then lets go of both the inorganic phosphate and the ADP. It's attached to a subunit closer to the plus end. Power stroke changes its shape, moving myosin to the right, effectively moving the actin to the left. Repeated over and over and over again in the presence of ATP, the myosin motor protein will be walking along the actin filament. All of those myosin heads at the same time. Think of how fast that has to be, causing contraction, shortening of the sarcomere. All of those sarcomeres together in all of those individual myofilaments will result in the contraction, the shortening of that muscle fiber. All right, we've run out of time. I will stop here. We will pick up here on Wednesday. We'll start with a clicker question, and then we'll talk about how this is regulated. How is this muscle contraction regulated? It needs not only ATP, but also needs to be regulated in response to nervous stimulation. All right, I have chat hours today at 12:30 if anybody has any questions for me. Otherwise, take a look at your SmartWork videos, and we'll continue with muscle contraction on Wednesday. See you later.